Welcome to the third video on viruses. So we're going to continue our discussion on how viruses are classified. Okay, so we talked about host range and size in the last video. So those are two ways that viruses are classified. So remember, just as a brief recap, viruses are measured in nanometers typically, although sometimes we do report them in micrometers as well, micrometers, my apologies. But fun fact, viruses are about 1/10 to 1/3 the size of a bacterial cell, because they are pretty tiny. And then host range, as we talked about in the last video, is that spectrum of organisms that a virus can infect, okay? And it is a very specific process because a structure on the virus must be complementary or a similar shape to a receptor on the host cell, okay? So those were the two things we discussed in the last video, which are two ways that a virus can be classified. This video is going to focus on the one other way, which is going to be structure. And then we'll have another video for the fourth way, which is their replication cycle. The notes say life cycle, because that's just a general term that we use for when something kind of starts an infection and then when it ends it by making new things, and in this case, new viruses. I'm a little hesitant to use life cycle only because viruses aren't alive, and I feel like it's just a little misleading. But life cycle is just a general term, that we use for how something goes about making more of those things. Okay, so let's get to the viral structure talk. So the basic structure of a virus, okay, at the most simplest viruses that exist are made up of two components. So all viruses are made up of at least these two components: nucleic acid genome and a protein coat called a capsid. Okay, and so this is what all viruses have, the bare minimum: nucleic acid genome and a protein coat that surrounds it and protects it. And that's, again, called the capsid. The capsid is made up of individual capsomeres, okay? And so when you're looking at the picture on the left, for instance, you can see the purple in there is going to be the nucleic acid, and I'm just going to abbreviate that NA, nucleic acid. And there's a lot of variability with the type of nucleic acid a virus can have as its genetic material. And then this entire blue structure as a whole, that's the capsid, okay? That's the protein coat. But if you look, each of these little individual circles, I'm just gonna highlight a couple of them, those are the capsomeres, okay? Capsomere. Okay, so the capsomeres make up the capsid. Now, some viruses are more complex, and some of them have what's called an envelope, which they actually take from the host cell as they leave the host cell. So we'll talk more about that in a minute. But a complete infectious viral particle is called a virion. Okay, so again, for those simple viruses, that's just going to be the nucleic acid and the capsid. But if it is a more complex virus, then we're going to go ahead and include that envelope. But whenever it's fully formed and able to infect a host, we call it a virion. Those viruses that do not have an envelope, we tend to refer to them as naked viruses, okay? Excuse me. And one thing I do want to mention is that viruses, as far as we know, are unable to go from being a naked virus to an enveloped virus. Okay, it's one or the other. Either it is with an envelope or it is without. Okay. All right. Let me change slides here. So this is where, honestly, viruses just blow my mind with how diverse and therefore complex they are. And this is one reason we have such a hard time studying them and combating them is because they're just so diverse. Okay, people tend to think, oh, well, HIV and influenza and corona and rhinoviruses, they're all the same, right? They're viruses. They have to act exactly the same, kind of like humans do, right? You subject two humans to similar conditions and their body is going to react the same way. We need the same things to live, right? And our bodies react the similar ways to, trauma or positive environments or whatever it may be. Viruses are not that way because they are so diverse from one another. So they're a little harder to figure out. So in terms of nucleic acid, some viruses have DNA, others have RNA, okay? But even taking that a step further, if you look at all the DNA viruses, some have single-stranded DNA as their genetic material, others have double-stranded DNA, right? Remember, all living things have double-stranded DNA. So those ones are most similar, but again, still very different than living things. And then if you look at the RNA viruses, things get very complicated. So with RNA viruses, they can have single-stranded RNA genomes. They can have double-stranded RNA genomes, which is interesting because all the RNA that's present in living things, it's only single-stranded. So it's kind of crazy that some viruses have double-stranded RNA for their genetic material. And then if you even take a closer look at those single-stranded RNA viruses, there's two different forms of it. As we'll talk about in a future video, nucleic acids have a directionality. They're the two ends of the molecule look different. And so we call one end the 5' end and the other end a 3' end. And I'll show you how we write that because you will see it again when we talk more about DNA replication. So we have a 5' end and a 3' end when we talk about nucleic acids. So some single-stranded RNA run 5' to 3', others run 3' to 5'. But because we're talking about viruses, we usually call this 5' to 3', we call that the positive sense strand. And then the opposite version, we call the negative sense strand. And why this is important is because this 5' to 3' is what our ribosomes can use to make protein. So remember, the virus takes over our cells and uses our ribosomes and our energy and our amino acids and things. And so viruses that come equipped with positive sense RNA, that can automatically be used to make protein. But if the virus has the negative sense, there's just extra steps in there to convert the negative sense to a positive sense. That way the ribosomes can use it. So it's, like I said, it's very complicated, but it's also really interesting how diverse they are. Okay, and even going farther than this in the diversity, their nucleic acids, some viruses have circular and others have linear nucleic acid. If you look at this picture, look at influenza virus on the top left, it actually has 8 separate RNA segments, okay? Which is super interesting, right? It has multiple nucleic acids that make up its genetic material. HIV has 2 RNA segments, whereas if you look at hepatitis B, it's got 1 circular DNA that serves as its genome. So much diversity. And this means how the virus replicates is going to be different. So all of these, the 3 on this screen, have very different replication cycles. All right. So next, if we look at the capsid, okay, the morphology or shape of the capsid, there are 3 big categories: helical, icosahedral, or polyhedral. I normally use the term icosahedral, and complex, okay? So remember, the envelope is something that's outside of the capsid, so I apologize if that 3rd bullet is misleading. So please note there are 3 capsid morphologies. So the helical viruses, their capsid is kind of like a slinky. If you've ever seen a slinky, right, when you stretch it out, it's very helical. But when you push it together, it looks just like a normal solid cylinder. And so that's what the helical capsids look like. And for the polyhedral or icosahedral, that's what you see in this, the top part of this bacteriophage. So they tend to look like this, like a hexagon. And so all the capsimeres are arranged in triangles. So I'm sorry, I can't draw this very well, but they'd be arranged kind of like this in triangles. And so it'd be a 3D shape. And you can still see that in the bacteriophage head. Although we say bacteriophage, which are viruses that infect bacteria, have a different type of capsid morphology. We actually call them complex because they have more than just the capsid. They have this sheath and the base plate. So our complex structure is when viruses have like this helical tail portion attached to the head region. Okay, so bacteriophage actually have the complex structure, but there are some viruses that just that they look like this. Okay, so this is what a polyhedral or icosahedral virus would look like if that was all there was to it. All right, remember we said some viruses are enveloped, okay? And an envelope is actually part of the host plasma membrane that the virus takes with it as it leaves the cell, okay? So in this top left image, you're looking at it like it's kind of like a time lapse. So the virus starts out, right, without having anything on its surface. And as it pushes its way through the cell membrane, so this here is the cell plasma membrane, right? It's a phospholipid bilayer. So as it pushes its way out of the cell, it takes part of the plasma membrane with it, okay? And that is the envelope, okay? And so we call this process of pushing itself out of the plasma membrane, we call this budding, okay? And when the bud pinches off, okay, the plasma membrane reseals behind it. So this form of exiting the cell does not kill the cell, okay? So lastly, I want to talk about diversity in the, it's not letting me. There we go. Diversity in glycoprotein spikes. And so, excuse me, oh, I got ahead of myself. Why do viruses actually want to take part of that host cell membrane with it? What advantages does that have? Well, by taking part of the host plasma membrane, the virus is essentially covering itself in host cell material, right? So it's a way to kind of camouflage from the immune system because it's trying to look like it belongs. It looks like it's part of the cell. And so it kind of acts like a little cloaking device here, if you like Harry Potter, an invisibility cloak. But of course it doesn't actually make it invisible, but you know, where it's trying to kind of hide in plain sight. The envelopes also help viruses go out and infect new cells because this allows them a unique method of entering a cell, which is called fusion. And we'll talk more about that in a future video. A big disadvantage of the envelope is that they are much more easily susceptible to the physical and chemical antimicrobials because it's made up of a phospholipid bilayer. And so as you can see in the picture on the far right, COVID has an envelope. And so that's why they say you can spray down like your workstations and other surfaces in your home and stuff, and it will break down COVID because that lipid membrane is easily, more easily destroyed than some non-enveloped organisms. So the other thing is that some of these viruses that have spikes, glycoprotein spikes, and these are actually the Achilles heel of the virus and how our immune system will actually go about detecting them, which is pretty snazzy. So even though a virus, hold on one second. Sorry. Even though a virus is taking part of that plasma membrane with it, it still has those glycoproteins that stick out and, help the protein attach to the surfaces of other cells and things like that. So our immune system is still able to recognize that the virus is foreign because of those glycoproteins. All right, y'all, that's the end of this video, although it's not wanting to let me exit, so that's fun. All right, let me know if y'all have any questions.